Sweet. Yeah, so I'm a postdoc working with Ian Wright as part of the centre at the Western Sydney nodes at the Hawkesbury Institute for the Environment, and working with Rachel Gallagher as well. And I started that postdoc about a month ago now. And for my postdoc work, we're going to be looking at adaptations to water and heat stress in eucalypts. But I won't be talking about my postdoc work today, but I am really looking forward to catching up with everyone at the retreat and talking about some of our ideas and how we can work with some others across the centre and in different nodes, for example. But today I'll be talking about some of my PhD research, which I submitted at the start of April, and that was at the University of Tasmania, working with Mark Hovenden and Tim Brodrube as well as part of the centre. And that project was looking at neighbourhood effects in a temperate eucalypt forest broadly. But today I'll just be talking about two chapters that made up of that PhD. So the first paper that I'm going to talk about today is canopy damage during a natural drought depends on species identity, physiology, and stand composition. So as we all know, being part of the centre, the climate change is causing warmer and drier weather, and this is really increasing the frequency and intensity of droughts. So drought conditions cause or place significant pressure on plants, and these can be stressful conditions for growth, and they can cause drought damage or altered growth rates, and in extreme cases can cause mortality. An increased tree mortality has already been observed across forest communities globally, and this has been widely reported in many studies around the world. And it's widely accepted that the primary cause of tree death during drought is failure of the water transport system due to xylem cavitation. So starting with just a quick overview of how xylem cavitation actually occurs. So in this figure here, you can see that water moves through plants from the soil to the leaves in pipes called xylem conduits. And then this water is lost from those leaves via transpiration. So this creates a negative pressure gradient through the plant, which pulls the water up through the plant via tension. But during drought, this tension can increase, and if it becomes great enough, it can seed air bubbles into these xylem conduits, which block the flow of water, and this is called xylem embolism or xylem cavitation. And these embolisms in these xylem conduits can either damage the tree or can cause tree death. And the vulnerability of different species to xylem cavitation varies greatly between different species. And so commonly to compare between different species, often this threshold is used when 50% of the xylem area is blocked, and this is called a species P50. So on this figure here, you can see this shows three vulnerability curves for three different hypothetical species represented by the different colours. And on the y-axis here, you can see this percent loss of xylem conductivity or percent of xylem area that's blocked by these embolisms. And then on the y-axis is negative water potential, so increasing drought or water stress as you move from 0 to 10 megapascal. And the earlier that these vulnerability curves cross that 50% threshold, the more vulnerable that species is to xylem embolism. And so this has been scaled up to explain mortality patterns or predict mortality patterns in the field. So, for example, in this hypothetical example, we would expect plant A to die first because it's the most vulnerable, followed by plant B, and maybe plant C survives because it's the least vulnerable to embolism out of these three. And this approach has had success in explaining natural drought damage patterns before, but it does leave a substantial amount of variation unexplained, which suggests that we're missing part of the story. And this may be due to the fact that this approach focuses on individual plants and ignores the effect of neighbours. So as plants are transpiring water and removing it and losing it as losing this water, they're also removing it from the soil, which is making it unavailable to neighbouring plants in close proximity. So if we stick with this same hypothetical example as before, if plant B was surrounded by members of species A, because species A is quite vulnerable to xylem embolism, relatively early on in the drought, these neighbours will stop using water quite early, and that means that they will stop having an effect on the soil water availability and will not influence plant B too severely. But if, on the other hand, plant B was surrounded by plants of species C, because these are more resistant to xylem, have more resistant xylem, they're able to continue using water for longer into the drought, beyond a point where, and draw down the water beyond a point which could potentially kill plant B. So this means that in a forest community, it's not just the density of neighbours, but also the identity of neighbouring plants that is important in determining the variation in soil drying across the community. So to address this gap, we conducted a drought damage study in the Australian Forest Evenness Experiment, or the AFEX. And this was my study site in my PhD, which was established by my supervisor, Mark Hovenden, and others in April of 2013. And this, the AFEX is located in the Lower Styx Valley in southeast Tasmania that you can see here. And I'd just like to pay my respects to the traditional owners on which this land, or on the land on which this experiment's located, and pay my respects to elders past and present. And this photo here just shows where the site was established. So it was clear felled a few years earlier and then burnt in a high regeneration fire. And the diagram on the right here just shows the site layout. So each of these squares here is a 25 by 25 metre plot, and the different colours represent 4 different replicates. And it was set up as a full orthogonal evenness by dominant species experiment, which used these 4 locally abundant species. So there was Eucalyptus regnans, Eucalyptus delegatensis, Acacia dilbata, and Pomaderus apetala. And these were replicates. These 4 species at 5 different evenness levels were created 20 different treatments, and this was replicated 4 times. And this photo here just shows the same area as what you were looking at before, but in March of 2019, so 6 years later. And you can see that there was substantial growth across the site, and even more since then in the last few years. And the AFEX just provides a really ideal opportunity to explore the role of neighbourhood composition and neighbourhood effects on tree growth and tree survival, because abiotic factors are held relatively constant across this site, so similar soil type, similar, all experience the same climate, etc. But there's vastly different community compositions between the different treatment plots. So this means that the effect of neighbours can be more easily isolated and teased apart. So we address these 2 research questions. So firstly, does natural drought damage correlate with species vulnerability to cavitation? And then secondly, how does the density and identity of neighbours impact drought damage? So there was a severe drought that occurred in the site over the 2018 and 2019 summer, and it caused substantial damage across the site. So to capture this damage and to quantify it, we established 14 20-metre transects, which were inside the main treatment plots. So these are just shown here by the yellow lines. And again, these white squares here are those 25 by 25 metre treatment plots. And the transects were 20 metres long and 2 metres wide. And then within that transect area, we recorded the species identity and the % damage of every plant in the site, in that transect area. And we also then calculated the density of each species as well. So we also determined each species vulnerability to cavitation using the optical technique. So in this figure here, this just shows the same as the hypothetical example as before. So on the y-axis, you have this percent loss of conductivity and that 50% threshold indicated by the dashed line, and negative water potential on the x-axis. So again, the later that they cross that threshold, the more resistant that species is to xylem embolism. So you can see here that Eucalyptus regnans was the most vulnerable to cavitation, followed by Delegatensis, then Pomaderus opetala, and then Acacia dilbata. And this correlated fairly well with the damage that we found in the transects. So this figure on the right now is just plotting the those mean P50 values, so these values here on the x-axis, against the mean percent damage on the y-axis. So you can see that both eucalypts suffered substantial damage, with Eucalyptus delegatensis suffering the most, around 35% mean damage, and Eucalyptus regnans about 25% mean damage, and Pomaderus and Acacia were relatively not damaged at all. So this means that this P50 trait and vulnerability to cavitation did correctly identify that the two eucalypts should be most susceptible to drought damage, which we saw. But we also saw that Delegatensis suffered significantly more, a significantly higher level of damage than Eucalyptus regnans, when it was actually slightly more resistant to embolism. So this is where the effect of neighbouring plants comes in. So to determine the effect of neighbour density and identity on drought damage, we analysed both species, the two eucalypt species separately, and we looked at how the damage in each eucalypt species was impacted by the density of each species as a separate predictor in the model. And this figure here just shows the model coefficients for each predictor in determining canopy damage, in Delegatensis in these black points here, and in Eucalyptus regnans in those grey points. So you can see that of the four species, only Pomaderus had a significant effect on the damage in both eucalypts. And you can also see that this was stronger and clearer for Delegatensis. So this gives evidence that the density of Pomaderus is potentially the cause of the greater damage that we saw in Delegatensis as opposed to Eucalyptus regnans. And so this does support our hypothesis that the strength of the effect of neighbouring plants is related to their xylem resistance to cavitation, because as you can see here, Pomaderus was significantly more resistant to cavitation than either of these two eucalypts. But we also should have theoretically seen an even stronger effect of Acacia dilbata, because this species here was by far the most resistant to cavitation, so it should have had the I guess the strongest effect on soil water availability. But Acacia is relatively rare across the site and relatively rare in density surveys because it had quite low germination success compared to the other three species, and especially compared to Pomaderus, which occurs at quite at very high densities because it's germinated from the seed bank as well. So it's possible that at these low densities for Acacia, we just weren't able to detect an effect. But why was this impact? Why did Pomaderus impact Delegatensis more than Eucalyptus regnans? We think that this was likely due to differences in rooting depth. So this figure here just shows pre-dawn water potential on the y-axis, which is a proxy for rooting depth, because pre-dawn water potential is when the root is at equilibrium with the soil surrounding it. So it indicates the availability of soil water surrounding that root. So therefore, after a period of dry weather, the less negative water potential, pre-dawn water potential, means that the plant has greater access to soil water or deeper soil water reserves. So the points here just show the mean and standard error in black, and the grey points show the raw data for 10 plants of each species. And you can see that there's overlap between all of these plants, all of these species, and they weren't significantly different, but it does show that Eucalyptus regnans does tend to be able to access these deeper soil water reserves, likely deeper in the profile. And so there's less overlap with the other two species compared to when you compare, say, Delegatensis and Pomaderus. Another possible reason that we thought that may account for this may account for the differences in damage rather than just the neighbour density. We thought this may be due to differences in stomatal behaviour. So, for example, if Eucalyptus regnans closed its stomata much earlier, so had a more conservative strategy during the drought period, it may have reduced the likelihood that it would have reached these water potentials that caused xylem embolism. But as you can see here, on the y-axis, so this is relative stomatal conductance against leaf water potential on the x-axis. You can see here that all three species sort of start closing their stomata at a similar point, about negative 1.5 megapascal, but you can also see that Eucalyptus regnans actually has a slightly more risky strategy than the other two species and closes its stomata slower, which is the opposite of what we would expect if this explained why it had lower damage. So this just means that the differences in stomatal regulation did not explain the differences in damage among the two eucalypts. So just to summarise what we found, so we found that vulnerability to xylem cavitation did broadly explain the damage patterns, but not completely. So damage was also the result of interactions among neighbouring plants, and specifically with neighbouring plants that had more resistant xylem that occurred at really high densities. So these results just really highlight the need to consider both the physiological and ecological processes to accurately predict the effects of future drought on plant survival. So for the second half of this talk, instead of drought damage, I'll be talking about eucalypt growth instead, which was another chapter in my PhD. But the premise of this chapter really follows the same reasoning as for the drought damage study. So in a warmer and drier climate in the future, how will potentially the strength and direction of neighbourhood effects on plant growth be impacted, and how will these differ? So this chapter on eucalypt growth is really focused in these mapped neighbourhood plots. So these were set up by one of Mark's honours students, Mel Gerwin, in the winter of 2018. And there was 114 of these neighbourhood plots that are spread across the site. So this just again on the left here just shows that the same site layout with those 25 by 25 metre plots represented by these squares. And then the red and black circle indicates a neighbourhood plot, which is surrounding either a Eucalyptus regnans tree or a Delegatensis focal plant, which is represented by the red and black colours. So each of these neighbourhood plots, which is zoomed in on here on the right, just shows the focal plant in the middle here, and then all these other points around are neighbours, and we mapped all these neighbours within 2 metres of the focal plant. And so for the focal tree as well as all the neighbours, we measured the height and the DBH in the exact location to an accuracy of about 10 centimetres. And then in total, we mapped and measured nearly 8,000 plants spread out across all these 114 neighbourhood plots. And these ranged in density from some were about 10 plants in total to others that were about 170 plants just within this 2 metre radius. So really high densities in some. And this also ranged in composition as well. So some were essentially eucalypt monocultures, so all just one species, to others that had a nice range of all 4 species. And again, I just want to make the point that it's due to the experimental design and the way that the site is, laid out, you can see that there's different neighbourhoods, so vastly different neighbourhood compositions, but they're very, very close in distance, which just means it's very similar abiotic conditions, which means that it's more easy to tease apart these neighbourhood effects. And so we remeasured these plants in each of the next 3 winters. So we have 3 years of annual growth data, and we recalculated the neighbourhood effect based on these new measurements when we remeasured those 8,000 plants, those 8,000 neighbours. And in addition to those mapped neighbourhoods, we also measured 35 isolated focal trees. So these were trees that were scattered around the site that we just naturally found that were had no neighbours within that immediate 2-metre radius surrounding the focal. So just like we did with the non-isolated focal plants, we measured their height and their diameter in 2019, 2020, and in 2021. So we have 3 years of annual growth for these trees too. And these images here just show examples of 2 focal trees growing with neighbours on the left here, and then focal trees without neighbours within 2 metres on the right here. And so using these isolated focal trees are a really ideal way to assess neighbourhood effects, because we have these isolated focal trees really sort of act as a baseline or a control of how these trees would grow without the influence of neighbouring plants. So first off, how did the growth rates compare among species, years and trees with and without neighbours? So this figure here just shows the mean standardised basal area increment on the y-axis, which is just essentially a log transformed relative growth rate, which accounts for differences in sizes between initial sizes at the start of the sampling period between each of the focal plants. And so these white points here show the mean standardised basal area increment. Of this mean standardised basal area increment for the isolated focal plants, and the black points here show it for the non-isolated focal plants. So for Eucalyptus delegatensis on the left, and Eucalyptus regnans on the right. So you can see here that there's a substantial difference across all years and across both species between the focal trees that were growing without neighbours and the focal trees that were growing with neighbours. So this just indicates that neighbours are having a strong and competitive effect on the growth of both eucalypts. But you can also see that the strength of this effect varies between the years. So for example, the difference between the black and white points as you should go from year to year. And so our next step was to determine what aspect of the neighbourhood was driving this competitive effect. So to do this, we used a neighbourhood index, which was the sum of all neighbour plant sizes. So each of the neighbouring plants divided by their distance from the focal plant. So this just scales the effect of each neighbour with the assumption that closer plants will have a stronger effect. And we calculated this neighbourhood index at different levels. So first, we assumed that the effect of all neighbours was equal regardless of their species identity, which meant that we had one neighbourhood index term for the whole neighbourhood, and we called this the community index. Next, we calculated neighbourhood indices according to their functional group, so whether they were a eucalypt or a non-eucalypt, so also the non-eucalypt being Acacia dilbata and Pomaderris apetala. And this meant that we then had 2 different neighbourhood index terms. And then finally, we calculated separate neighbourhood index terms for each species individually. So this meant that if neighbouring species were having differential impacts on eucalypt growth, like what we saw in chapter 1 with a strong effect of Pomaderris, then we would detect this in the more complex model where we have these 4 different neighbourhood index terms. So we fit these multiple models and which differed in how we grouped that neighbourhood index, and we used model selection to determine which level best described growth. And so we found that in both eucalypts, the community level neighbourhood index was best. And this suggests that all neighbours, regardless of their identity, are having a similar effect on eucalypt growth. So this top figure here shows the same standardized basal area increment as before on the y-axis, or absolute standardized absolute growth on the y-axis, and then the community neighborhood metric on the x-axis. And the different years are plotted in different colors. So black is 2018, 2019, red's 2019 and 2020, or that growing season, and the blue is 2020 and 21. And you can see that the strength of this relationship really varied substantially between the years. So being strongest for both eucalypts in that first year, represented here by the black line, and weakest in 2020 and the 2020-21 summer, represented here by the blue line. And the two diagrams at the bottom just show examples of neighborhoods with a low on the left, low community neighborhood metric, and a high community neighborhood metric on the right. So you can see that in both eucalypts that as neighbours got bigger and closer to the focal tree, the growth decreased, and this was particularly evident in that first 2018/2019 year. And this just supports the previous figure, which I've just brought back here, where you can see the difference between plants with and without neighbours. So you can see here that this difference decreases in that second year slightly, and in the third year a lot, and particularly for Eucalyptus regnans. There's very little difference in that final year, which indicates that neighbours are having very limited effect on regnans growth, which you can also see in this blue line up here where it's almost flat. And so our next step was to determine what was driving the difference in strength of competition between the different years. So we calculated a few different climate metrics, so total precipitation, mean temperature, and the standardized precipitation evapotranspiration index, which is just a drought index. And we calculated these either over the entire year-long sampling period, as well as just the growing season, so from October to March. And we found that the growing season metrics were well correlated with the strength of the competitive neighbourhood effect. So this was true for both total precipitation and the drought index. But we didn't see annual conditions have a clear relationship with the strength of the competitive effect. So this bottom figure here just shows the slope of each of these lines in the figure above on the y-axis. So here a more negative number in this figure down the bottom indicates a stronger relationship, so stronger competition. And you can see here that as the amount of growing season precipitation increases, the strength of the competitive effect decreases, and this holds true in both eucalypts. So this really suggests that neighbour interactions for growth are strongly dominated by competition for water in this system. And when it's more limiting, the competitive effect of neighbours is stronger. And you can see in that first year, so this is the same indicated here by that black point. So this is the same year that there was the drought in the previous study that I talked about. And you can also see that so you can see that this was the year that the competitive effect was strongest, and this was really true for delegatensis, which had a much more negative, so a much stronger competitive effect in that first year. So this just supports our previous finding that delegatensis seems to suffer more from greater competition for water. So just in summary, we found that neighbours had a strong and consistent competitive effect on eucalypt growth, but we didn't find the species identity of neighbours to have a strong effect. We found that the competitive effect varied in strength among years, but in a relatively predictable way. So the drier climatic conditions, there was stronger competition, but in wetter climatic conditions, there was weaker competition. And again, we just saw the Eucalyptus delegatensis suffered more from competition than Eucalyptus regnans, and this was in growth, but also in the drought damage study. And this just really, again, just suggests that neighbour interactions are dominated by water competition in this study system.